Hello, everyone, and welcome. This is Dr. Fox, and let's talk about laboratory safety. It is of the utmost importance that we maintain an environment and culture of safety so that the laboratory is a place where learning may occur at a high level and individuals will feel safe to be and to work. An important part of safety is wearing personal protective equipment, or PPE. Some of the PPE will be provided, and some we ask that you bring yourself. So let's start with what is provided. All M1s will be provided with a laboratory coat to use in the labs. These lab coats are typically laundered around the winter break, or if they become amazingly dirty or unusable, we may offer a replacement. Nitrile gloves for your hands are available in all of the labs. We ask that you try to limit how often you change your gloves. So, amidst dissection, it might be a good idea as needed to wash your hands with gloves on so as to not have too slippery a grasping surface. To protect your eyes, we'll provide you with a pair of ANSI safety glasses. If you are like me and you wear glasses, we have pairs that fit over existing glasses, although these might not be compatible with all fashion frames. So if you have a particularly large-sized frame, it might be to your benefit to explore a pair of glasses that fit you and that allow you to operate comfortably in the laboratory. I recommend going to a local hardware store with your glasses and trying on a number of frames. Just make sure that they are ANSI certified. Also provided will be procedural masks for times when we are cutting bone so that we don't inhale any bone dusts or particles. We also ask that you bring your own closed-toed shoes so you're not wearing slides or sandals into the laboratory, which would be a hazard. And also long pants. This should keep your lower limbs completely covered and safe in the laboratory. I know this sounds a little corny, but we ask that you come with a healthy dose of common sense. If it seems like it would be dangerous or hazardous, we ask that you don't do it. And if you see someone doing something dangerous or hazardous, we ask that you ask them to stop. And if you're not comfortable doing so, please just point them out to an anatomy lab faculty, and we would be very happy to intervene on all of our behalves. Communication is something that really isn't brought or worn, but it's an important part of the experience. So if you're feeling uneasy or unsure about something in the laboratory, whether it's the location of PPE, if you're wearing the PPE correctly, how to clean a particular instrument, how to load or unload a scalpel blade, or maybe you're just feeling a little unwell, I would encourage you to talk to at least your lab group, if not your anatomy faculty, so that we can help you navigate through these issues and make sure that you are on a path of safety and success. The safer you are, the better everyone will be. One of the bigger hazards in the laboratory are loaded scalpels. So here we can see scalpel handles with blades attached. Without the blades attached, the scalpel handles are very excellent instruments for blunt dissection. This edge in particular is very good for separating tissue layers. With the scalpel blade attached, you have an instrument that excels in cutting. We will rarely use scalpels in the lab, although they will be present for your use, and predominantly they will be used for removing donor skin. People typically get cut with a scalpel, either loading or unloading blades. Within the orientation, your faculty will show you the proper and safe way of loading a scalpel blade and unloading the scalpel blade. If this is something that you ever want a reminder of, please don't hesitate to ask your faculty for help. We are there to keep you safe and to aid in your learning. Now, for removing scalpel blades, we have a very handy device known as a Click Smart. These are affixed to the walls at the end of each laboratory adjacent to the sink. And you'll notice there is a little cartoon of the proper orientation of the scalpel handle and blade that fits into this slot. You advance it until you hear a click, and then you withdraw the handle, and you are ready to either wash and put away that scalpel handle or reload it with another blade to continue. When using scalpels, it's important to make sure that you're working with a sharp blade, and it's also important that you pair that instrument with another instrument such as forceps or needle drivers, hemostats, so that your other hand that isn't involved in driving the scalpel is holding the tissues in a safe manner so that you're not cutting yourself. Donor care is something that we take very seriously in the laboratory. It's an important part of honoring our donors, and it's also an important part of keeping everyone in the laboratory safe. Students are not the only people that encounter donors. Faculty do as well. There is anatomical donations staff, and any of these individuals may unshroud your donor at any time. So it's important that we keep the tables clean. And what does that mean? Well, it means that any waste products, whether they be gloves, paper towels, scalpel blade wrappers, they're going into the white waste barrels. Red waste barrels are for unembalmed tissues, so we really won't use them in the duration of M1 anatomy because our donors are all embalmed. Any donor tissues go in the fiber barrels that rest underneath the donor table on that shelf. It's important that that fiber barrel remains on that shelf, that you don't put it on the floor or any other surface, and you maintain the lid on it at all times. With respect to donor care, you're looking at a donor that is under a plastic shroud, and underneath that plastic shroud, the donor is surrounded by moistened muslin. And that muslin tissue is soaked in a substance known as biostat, or cadaver hydrating fluid. You can find biostat in the sinks, typically kept in these smaller carboys. They should be labeled. Or we also have biostat in yellow spray bottles in the labs, also labeled. Biostat is something that you will apply to the muslin that covers your donor, and not typically the donor directly. It's something that you want to use sparingly, hence we have it in the yellow spray bottles. We don't want you to use it generously because an overuse of this tends to create mold in the long term. Another substance that you may see out in the laboratory is monoethanolamine, or MEA for short. This is for faculty use only, and you'll see it in a red spray bottle. This is for the neutralization of formalin. So formalin is a laboratory hazard. Your donors have had MEA circulating through them to help neutralize the formalin. Sometimes as we dissect into deeper and deeper spaces, not all of that formalin has been neutralized. And so if you ever encounter a formalin smell, or if you notice that your eyes or your nose, any of your mucous membranes are burning, that may be formalin. Just contact a faculty member and they can use MEA to help neutralize that in your donor to help keep you safe. Now, whenever you're not working with your donor, it's recommended that you keep your donor covered. Certainly when you put away your donor after you're done dissecting, you should clean all of the materials from the table. Any waste products go into the waste bin. Any donor tissues go into the fiber barrels. And then you can use Sanazol 7, which I will discuss in the next slide, to cover any hard surfaces. Once that's complete, you can cover your donor with the muslin shroud, tuck it in around all of the edges of the donor, and then cover the entirety of the table with the plastic shroud, such that all four corners are covered. This will lock in the moisture for the table and help preserve your donor such that they will last for the duration of the course. Let's continue this discussion more in the context of a clean table. So the cleaner the work area, the safer the work area. So as you work, please endeavor to keep the area as clean as possible. Dissection can be a messy endeavor. I would not recommend having lots of waste product on the tabletops. So as you generate waste, please do deposit it in the white barrels throughout the laboratories. Please keep your dissection instruments in the kits. When you're using it, they may be on the table. Sharps should always be in an area where they can readily be identified so that they don't pose a threat to anyone. As you remove donor tissues, you may opt to keep them in a small pile on the tabletop, and as that pile grows, you can move them from the tabletop to the fiber barrel. Fiber barrels, again, remain on the donor table in that lower level. We don't put them on top of the donor table. We don't put them on the floor. We don't put them on the countertops. And this is because the floor will oftentimes have mold spores on them. And if we transfer mold spores from the floor via the fiber barrel or anything else to the donor, this really endangers the donor. They may begin to have an outbreak of mold, which can really lower the usefulness or make the donor wholly unusable for you. So let's try to break this cycle here and keep any mold on the floors. Sanazol 7 is a detergent that can be used to clean any fomite surfaces. So countertops, you can spray it on paper towels and wipe down computer mice or even keyboards with it, tabletops, instruments, etc. Spray a little on them, let it sit, and then you can either wipe it off with a paper towel or rinse it off with water. Instruments go back in the instrument kits, which are kept also under the table as well. Sanazol 7 is something that is very safe to use, therefore we have it in a green spray bottle. If there is an injury in the lab, the first thing that you ought to do is evaluate, is this an emergency? And if so, please do call 911. You don't have to wait for faculty or colleague approval. If you feel that the situation requires it, please do call 911. There is a phone mounted on the outside wall of Lab 2 if you need it, or you may call from a cell phone. The labs are room 3613 MedSci 2, and the address is 1137 Catherine Street. If you forget either of these things, signs will be posted for you. You could just say MedSci 2, and EMS typically arrives at that 1137 Catherine Street entrance. It would be a good idea to send a student out to meet them. At this point, if we are in a lab period, faculty will be facilitating all of this, and this isn't anything that you would have to worry about. But if you're in the laboratories outside a regularly scheduled M1 experience, this is something that you'll have to do. After everything has been handled and the emergency is no longer, after you have returned from the emergency medicine department, then you will be required to complete an illness or injury form. And this is something that your faculty members or myself and Dr. Alsop can help you facilitate. If the situation is sub-emergent, there is a process, and this is in line with the scientific trunk illness or injury process. If you're injured, you contact Occupational Health Services. The pager is 5356, or you may call them at 734-936-6266, pager 5356, and also contact your anatomy leads, Dr. Alsop and myself. There are emergency eye washes that are located in the middle of each laboratory by the sink, so you can turn that on if needed. There are Band-Aids that we have available by every sink, typically mounted above the soap dispenser. There's also a first aid kit in the hallway adjacent to the phone, and you can also find a folder of material data safety sheets adjacent to the phone and the first aid kit in the hallway. Now, these instructions are posted in every laboratory. We don't expect you to remember this, especially if there is an issue. So just look around, find these instructions and follow them. If it's during a typical anatomy session, it's something that anatomy faculty will facilitate for you. We appreciate students working with faculty to maintain an active culture of safety, and we are grateful when students ask questions and are very clear communicators regarding what they need and what they are observing in the laboratory, so that we may all help to keep you safe and support your learning. Thank you so much for your time. I look forward to talking to you in subsequent videos. I look forward to working with you in the laboratories, and I am grateful for your attention and participation and maintenance of our culture of safety. Have a great day.